Thanks to everybody for joining today. I'd like to welcome everyone to today's webinar with Dr. Waxman and Garry Nolan. We're thrilled to have Garry with us today. Gary's a world-class scientist. He's a member of the Stanford Cancer Institute and is extremely well respected at the NIH and in the world of cancer research. Garry recently joined the board of the Samuel Waxman Cancer Research Foundation. We're thrilled to have Garry, very lucky to have Garry, and he joins a great group of scientists who are leading the fight against cancer. I'm now going to turn over the webinar to Dr. Waxman, and we look forward to giving you a very thoughtful insight into how to leverage business with cancer research. Dr. Waxman?
Thank you, Michael. And hello there, Garry in California. It's great to have you join us today and discuss some of your work. I'll just say a few words about you that Michael started with. You are one of the more outstanding scientists at Stanford University, a chaired professor, a respected teacher, innovator, and someone knows how to translate science from the most basic form to clinical utilization, which has made impact in many things we do today in understanding cancer. I hope you'll illustrate today how complicated a tumor is. People think that a cancer is just one cancer cell, keeps growing and growing and growing. You have demonstrated and guiding us to give us all the knowledge that there are multiple types of cells within a cancer that makes the cancer cell have its way, educates the cancer cell, and helps the cancer cell. So with that kind of knowledge, we're learning not to just treat the cancer cell itself, but all of the other players within a tumor. And the way we do that is from getting the knowledge of very sophisticated imaging of a cancer itself. And that's what you have been providing us. You've also provided us with the inspiration that you can take basic science from the bench in a lab and bring it to the clinic. And you continue to do that, and you advise government agencies as well as companies on how to do that. And we're happy we were able to provide you seed funding a few years back for some of this work and enabled you to bring out some innovative research and also bring that to the formation of a company that we secondarily benefited from. As you made it happen, you gave back to the Samuel Waxman Cancer Research Foundation, which I really do respect. So that's a little bit of my introduction. I look forward to your presentation. We may have some time for questions. And folks, meet Garry Nolan.
Well, thank you very much, Sam, and thank you very much, Mike and Jesse, for helping put this together. So as Sam outlined, one of the complications usually of being an academic is feeling that your work isn't always necessarily right on the cutting edge of what can be used for the public. You're often taking money in the form of grants from the government or other charitable foundations, and you're always feeling that you need to find some way to give back other than just in publications. So certainly as you publish, other people can take advantage of the work that you've done, but sometimes and often the person who's best able to implement the technology is the person who believed in it so much that they were willing to spend years of their lives studying it. So I'm going to talk today then about how over the course of 20 or 30 years, I've learned how to do this with the setting up and or sale, the exit, as it's often used, the terminology of those companies, to be able to get them out to the public, to be able to show that they're useful, and then to be able to use those resources to bring back to either the charitable foundations or frankly, sometimes even to the students to help them see that all their work has benefit beyond just publishing things on library shelves. So I'm going to do that by giving examples of about 2 or 3 different companies that I've started. and show you how it is that I took them from an idea in the laboratory to commercialization. So in general, and I'm not going to go over all of this today, but it's really about, from ideas to products. And it's really about how do you manage the academic industry divide? Because at least when I started in science back in the late mid 80s to early 90s, it actually was almost forbidden for an academic to consider that they would commercialize their ideas. And so it was really something you needed to do very carefully because your other academic colleagues might look askance at you. So really it was about how do you, at least the way I looked at it, was how can I show people that this can be done in a fair way that respects everybody involved, including the students, so that the ideas can be taken beyond the laboratory and beyond, frankly, what the NIH is really set up to do. When do you decide that a laboratory idea no longer should be funded by the NIH, and when is it mature enough to take it outside for commercialization? So I'll cover a little bit of all of these, defining what should be done from what can't be done, right? That's the beginning of it. Pitching and funding the idea to the NIH in a way that focuses on the broad utility. The idea being here that you can have a technology idea and you need to be able to show that it's not just an idea for yourself, but it's going to be useful for many different things, right? Creating in the academic setting, the right kind of paper trail with the university so that when it comes to the point of patenting, implementation, and commercialization, you have all your ducks in a row so that it can move quickly. You have all the permissions, the deans, et cetera, are ready for it, especially if you're in a university that doesn't really do this on a regular basis, so that later on, nobody will question you from a legal standpoint as to what it is that you're doing. And as I said before, knowing when that idea needs to transition, knowing your VCs, all right, that's a whole talk in and of itself. Who are the people who are eventually going to fund you and what are their goals? Finding the right management and delegating that authority, knowing where your role ends, right? And frankly, as the company matures, knowing when your role is over, right? And that it's time to move on to create another idea, right? And to take another company forward. There's a point at which that maturation process happens, and sometimes it is a surprise to the founder that they're no longer needed. So knowing that stage is important to really understand. So a little bit of my background. I trained at Cornell, Stanford PhD, MIT postdoc. I have a number of papers across a broad range of areas, everything from genetics to bioinformatics and robotics. 33 patent families, across all of the things that we developed. And luckily here I was able to learn a lot about this from some of the founders in the field of patenting. The guy who patented the early Cohen-Boyer patent at Stanford, he was my first patent attorney, and so I learned a lot sitting at his knee about what the key elements of patents are. And then I've spun out 10 companies to date, and I'll just take you through a few of them, and then I'll focus on 3 of them. And all of this was done pretty much when I started as a professor at around 1992. Very quickly had about a 30-member lab and has maintained that size throughout and taught these students. Many of them have gone on to start their own companies now. And this really is the engine of innovation. 30 members and setting up what I call a permission structure for the people to be able to feel comfortable coming up with ideas on their own, and that I will take it forward for them or with them and teach them how to do that. And so now my laboratory actually often attracts individuals who might have an interest in starting a company because they know that I can show them really how to do it. So here's just a short thing about the company experience that I have. Everything from a company that went public many years ago. And the idea, the technology-based idea, was really design viruses that reverse evolution to find viruses that make drugs, that make cells better rather than sicker, and then discover how it is that they do it, and then from that develop a drug. One of the drugs that came out of that is now an FDA approved drug. I long ago exited the company because they didn't need me anymore. Probably the technology for which I'm most well known is this one called CyTOF that was developed by DVS Sciences. Actually, another professor developed it and came to me to help me monetize it or commercialize it, or at the very least, show how it could be used. And that was eventually sold for over 200 million to Fluidigm. And the technology base here was actually building on something that my first professor at Stanford had originally built, called the flow cytometer. Len Herzenberg at Stanford had built something that I was entranced with, and it had everything from computers to lasers to physics to cell biology all in one, but it pretty much topped out at what it could accomplish. And I mean, it's still used today, it's fantastic, but it could go further. And so I brought basically other technologies to take it further. And that's an immune monitoring platform that's used the world across now for deep profiling of blood and cells. Another company called BINA, which was a genome assembly, basically a computational program that used very fancy nanotechnology for basically computers. We sold that to Roche for 110 million. A number of other companies that, some of which I'll talk about now that I won't delve into it, but all of it started with a technology base, an idea that I thought would take science forward. And usually the idea that it took forward was because I felt we couldn't do something today that we really needed to do. And I felt, perhaps in a self-aggrandizing way, that if I could give this to people, they would be able to also go beyond what their current work is and the limitations of what the data was that it could be provided. And two of those companies that Sam alluded to, Apprise and Akoya, have both been commercialized and are now, and actually some of the money, some of the stock from that I gave back to the Waxman Foundation. And then, so I'll talk about some of these other ones, as well as other companies that I'm still now in the process of setting up. So how do you determine what it is that you want to do? How do you make something that other people might want that is sufficiently distinctive that you can actually perhaps start a company around it? So most of the things that I do are what are called technology platforms, right? That's different than a drug development. That's different per se even than a diagnostic, right? This is a broad platform that I think many will benefit by, and that in so doing, actually, you might be able to even mine the things that you produce to make a drug or to make a diagnostic, right? So how do you determine what it is that you should do? And that really is asking the question, what's next, right? What is next that needs to be done? And the answer, at least in my mind, is you need to define what is inevitable, right? What inevitably needs to be done, what inevitably needs to be known, right? That irrespective of what we can do now, we really want some level of more perfect information, right? So it's almost a philosophical notion. So what is inevitable, right? So when I look at it in terms of how I think of it, like what is inevitably needed in cancer research, we inevitably need to know more and more about the cells and how they are arranged in a cancer, how they relate to the immune system, et cetera. So right now, the way that we do it is very simple, right? In fact, the technology that is in most clinical laboratories right now is about 50 years old and hasn't changed since, practically Stone Age ideas. So when I look at data or when I look at the current state of knowledge, I often say, well, what could be better, right? I see what could be better. I said, oh, this is awful. I need more. I need to be able to do the following kind of technique more efficiently. How do I do it? So then the best way to do it is almost kind of a form of reverse engineering. You then imagine what might be the perfect solution, right? If I could do this, that would be amazing. Well, I can't do that because the technology isn't there, but you can begin to work backwards towards what is possible. And even if it is not what is perfect, what is possible is often better than what currently is. So when you've defined what is possible, then you start to search around for, well, how do I put together the pieces of other people's technologies or other ideas that might be out there to create that possibility, right? So, all of the technologies I'll show have really been based on that kind of an approach. So to do that, though, requires a background, right, in the knowledge of tools and methodologies. So as it turns out, when you look at and read a paper, people often will just read the results, right? What is it that they did? I'm more interested in how did they do it? And I've always been interested more in how do they do it. I actually almost have always first gone to what we call in science the methods section. Because if I don't know how they did it, I don't know how to interpret the result and whether or not it actually was done correctly, right? So did they use the right algorithms? Did they use the right tools, et cetera? But as it turns out that as you begin to build up a catalog of methods and approaches in your head, they almost start to self-assemble into new ideas, right? Because when you say, what is it that I want to do? Almost immediately in your head, you have a pipeline of opportune methodologies that you could put together one after the other or insert a new one as needed to accomplish a new outcome. And again, often many of the ideas that we came up with were not things that were so novel. It's that we took them from another field, right? Mechanical engineering or physics or data sciences, right? And then we applied them for biology in a way that biologists had never seen before, right? So it's all about bringing something else novel to the field to build something new. So the other key issue here, and this is, I think one of the most important things that I try to teach my students especially, is ignore the critics. You know, the old saying is, if an old professor tells you something can't be done, they're usually wrong. If they tell you something can be done, they're usually right. So ignore the critics, have confidence, but not arrogance in your idea until you prove yourself wrong, not that other people tell you that it's wrong. So as before, define what should be done from what you can't do now, right? So the first of these ideas was the unmet need was diagnostics, as I said before, are still done by 1950s technology. So we created a technology called Codex 2 that I'll talk about to vastly scale up the depth and refinement of the imaging that can be done on a slice of cancer. So for instance, when you go to the doctor and they don't like something, say, on your skin and they take a biopsy from you, they actually send it off to a clinical laboratory who will slice it up and they'll do different forms of imaging on that cancer to see what kinds of cells are there, immune system cells and cancer cells. And then based on certain criteria, they'll say, well, this is okay, or this is a bad outcome, you need further therapy. So the technology that I'll show you in a moment is now used in many dozens of labs worldwide, and we're actually heading towards clinical implementation of it. And importantly, and this comes back to how do you incentivize the people inside of the laboratory, I founded it with 2 postdoctoral fellows. Equally, we had equal shares in it, because I've seen many of my colleagues, for instance, who have tried to start companies, and they basically, some of them have said, well, this was in my lab, so therefore, and I'm the famous professor, therefore I should get more. Well, that's the best way to annoy the students who might have been actually the ones in the laboratory doing all of the work. So I think it's important to basically share and share alike. And it really overcomes any of a number of different obstacles. So the first thing we did was we obtained significant public grant support by demonstrating initial utility. This is the usual kind of approach you take with preliminary data. You go to the NIH, you show them that it works. Often, the better of a track record you have, the easier it is to accomplish that, but not always. You don't get a free pass. And then that is really where the beginning, frankly, of the guilt begins, right? At what point does getting money from somebody else load onto you the basically requirement to somehow give back, other than just doing the work that you said you would do, right? We often will end or begin even our grants by saying, how amazing it would be if we do this and this will do this or that for cancer research. Well, how about actually doing it? And as I said earlier, usually it's the person who did it first or who conceived of it first who really is the one who's best able to take it forward. So the important first thing that I do with almost all of the technologies, well before often we even publish it, is start talking about it openly, right? You're really seeding the field for the idea. You're preparing the field for the idea, and you're trying to get other key opinion leaders, right, in the area to say, wow, yeah, if I could do that, that would be great. And the way I often talk about it is that you end up being almost like an arms merchant, right? If you get into an area where you are trying to prove one mechanistic or one result versus another, very often what you end up doing is getting into a fight with the people who think it is the other, right? Rather, by developing what I call a platform technology here, you basically are going to give it to both sides, both of which will say, well, gee, if I can do this, I can prove my idea over the other person's idea, right? So you're really basically, in a very mercenary way, providing something that everybody can use. And hopefully in the first publications that you have on it, what you do is you implement and show that idea so that they say, oh, there's an example of what I could do. Wow, I'll just substitute what he did with what I do, and I'll get my next paper in Cell, Nature, or Science. And then they come to you, and the best way to do it is basically what we call open source science, the field calls open source science, where you share the technology and the ideas as quickly as possible. Because the best way to get somebody to ignore you is to not share it with anybody. And then, if the idea is that important, they'll, somebody else will invent it, or they'll invent a way around you, right? So share, own the niche, as I say, and then move forward. So here's one of the examples of what this technology could do. And so CRC stands for colorectal cancer, right? So basically colon cancer, right? And there's this whole issue right now in the field about how the immune system postures itself relative to where the cancer cells are, what kinds of immune system cells infiltrate the cancer. That's all very important. So what we did was we had taken, it's not, there we go, one more, sorry. So we had 750 patients, all stages of the colorectal, fully what we call clinically annotated. This database of samples goes back 30 years from Germany. And we culled it down to two kinds of case types, patients who did well and patients who did poorly. And this is just a terminology, CLR and diffuse, that is really more clinical. Here's the close-up of those two different types. This is called the CLR, and here, this is called the diffuse inflammatory infiltrate. This disorganized mess here on the right are actually the patients who do poorly, right? These are the ones who do well. Why? Right, I mean, we can see these features, but what is it about the immune system cells beyond just this black and white, which is the standard staining that, as I said, is the 50-year-old technology? We need to go deeper. We need to colorize this information. So the technology that we developed allows us to look at all the individual cells, as you see here, right? And so these individual cells, then each in the color that they are given according to the technology that we created, allows us then to call out what those cells are individually. Sorry, this is on some sort of an automatic. Call those cells out individually and numerify them, right? And once you get those numerically determined, you can create the kinds of spreadsheets and algorithms that anybody who might be in the stock market business understands, is that this is how you can predict then what outcomes are based on the relationships of either one stock to another, or how one thing is playing against the economy of another country. We're doing the same thing here. We're looking at how cells, in this case, the cancer itself here in blue, how it organizes itself relative to the different kinds of what we call neighborhoods of immune system cells, right? And so once you see what those neighborhoods are, whether they have T cells in them, et cetera, follicles, all the different subtypes of immune systems, we can then begin to look at these almost like a social network, right? So you understand that knowing who clicks on what advertisements tells you something about what that person is, and then who their friends are will further refine that information. The same thing is here in cancers, where we can look at the immune system cells in a given neighborhood. This purple is a neighborhood that has, say, 10 different kinds of immune system cells in it. And that will then predict for us what the outcome might be. And it gives us ways to think about the biology that's going on there and actually develop both therapeutics as well as diagnostics. And that work right now was just accepted to Cell in the last week, which is one of the top 3 journals for my field. And that will be coming out shortly. So with that kind of background, with that kind of result, that was sufficient for me to be able to go to venture capitalists to get the money to start the company. So the company is called Akoya Biosciences. Akoya means rare stone, gem, or pearl in Japanese, for instance. So we spent several years validating the technology, more than 2 actually. We filed the necessary patents, so we protected ourselves even before we started talking about it publicly. I started with my 2 students, an outside LLC, identified, as I said, the necessary VC partners. I told you this already. The important thing here, give to get, so that they feel that they're going to put in the necessary work. And then we built this instrument, which is what's being used by everybody now around the world. And we have actually a next generation one coming in the spring next year. We then did, to be able to actually sell it, just to go back to Stanford for a minute, I can't take an idea in my lab and just give it to myself, right? That would be self-dealing. Stanford won't let you do it. So there's actually what's called a public auction of the technology. The technology goes up on the Stanford website. They say anybody who wants this can come and do a bid on it, or can say what that is that they're going to do with it, right? What would be the diligence? So we started the public auction that goes on for 2 months. I put in my bid, others put in their bid. I won. And usually, frankly, the Stanford professor who starts it wins, because Stanford knows, again, and going back to this issue, the people who are most likely to understand it and implement it effectively are the people who invented it, because they really believe in it, right? So it really comes back to, even from the very beginning, believing in yourself and believing in the idea and believing that you know at least how to take it into early commercialization. We moved some lab people to the company. After starting the company within a year, we actually bought a division of PerkinElmer, about 30 million. We raised another 30 million because we saw that they already had a product distribution pipeline for a lower end version of what we were doing. So they already had all the contacts, et cetera. They were already in the clinical lab. So that allowed us to take this almost immediately into preclinical practice, right? To actually, and actually is being used now at everywhere from MIT, Dana-Farber, Harvard, et cetera, the world around. So because we saw that that would be a great way to increase the base technology and actually start with revenues, right? That's one of the biggest problems with startups is, when do you actually get revenues? And so we started almost immediately with that acquisition, enabling revenues. And as I said, it's used worldwide. So a second company that we started, which actually has a very similar objective to read the images from cancer biopsies, but using a wholly different technology base, is called IonPath, right? So it was a postdoctoral clinical fellow in my lab, who actually I thought when he first showed up in my office was crazy, because he had this crazy idea. But again, you learn, just as others I think learned watching me, when someone is so passionate about something and they explain it so well, and even though it's not possible today that you feel that they can implement it, you give them the chance. So gave him the chance. We spent 3 years validating the technology base. He brought in others to the laboratory. So one of the other things that I do, which is, as I said before, is giving people a permission structure. When postdocs start in a lab, I say, Your job is to build an empire in my lab. Don't hurt anybody, but your job is to convince other people to work with you, right? And by doing that, what I found is that that creates the ability for leaders to form, right? That they're not going to feel I'm going to come back in and veto them, except when necessary. I'm saying, look, learn, I'll help you, convince others, hone your skills. This is the place. Be whoever it is you want to be, not who you were, is usually the way I start the conversations with the new postdocs, at least. So just like before, filed patents, started an LLC, identified VC partners, did the technology auction, and took the company forward. So actually started it with 2 postdoctoral fellows and a graduate student. The graduate student is actually now the CEO. The company's about 4 years old. They raised an initial $12 million, and they're already 50 people, and they're funding on revenues, right, moving forward. And those 2 of the postdoctoral fellows were so successful in implementing the technology and then publishing on it. They're now both assistant professors in the Department of Pathology at Stanford, with their own flourishing research laboratories, and fully independent of me. They have, you know, basically I'm the granddad. They don't really pay much attention to my needs anymore, which is perfectly fine. And this is the instrument here on the right, and it uses an advanced form of mass spectrometry. We're shooting beams of ions at the gun. This looks like something, frankly, out of a James Bond film, right? James Bond would be down here, and the evil villain would be off on the side chuckling about what he's going to do. But so this is the technology here. And actually, it's much prettier now. It's in a nice, pretty box. This instrument here costs about $1.5 million, and frankly, they're selling quite well because of what it can do. People will pay for being able to do something that they can't do today. And now the final company that I'll talk about is something called Atom Naught. This is something I'm in the process of raising the funds for. It was the idea of a researcher from Australia who came to visit with me, and we cooked up starting a company around it. Same idea here, working now with Stanford to bring a relatively mature idea into Stanford in a way that gets around what's called conflict of interest, right? Where it might be that I have to give up any of my stock for this, right? To be able to work on it in my laboratory because it still needs a little bit more maturation. But the idea is to bring something which is so hot and so novel in what it can do. And this actually, what this technology does is it can image every single atom's position in a cell. Every atom with such resolution that I can tell the difference between a single bond and a double bond. And if you're a chemist or know enough chemistry, that's remarkable. There's nothing else out there that can do this. This technology can do it, right? So for me, being able to accomplish that is so exciting. And so, right, what's next? What can't we do? That I'd be willing to give up any of my stock for that. And here's actually, I'll give you a bit of a video of sort of what the technology does here. This is obviously an animation looking inside the device. I hope it comes across well enough. Your cell is sitting on the edge of this sample here, and we can literally decompose the cell atom by atom and pick up its exact positioning of where it was in the 3D structure, and then we build below the image of where that cell, where all the atoms were, and what their identity was. Was it nitrogen, oxygen, carbon, et cetera? And then from there, we look at really what the protein or otherwise was. And then we rebuild the structure of the cell. Nothing out there today can even come close to this. Closest thing is cryo-electron microscopy. And this will hopefully supersede even that. So from ideas to products, managing the divide. So as I said before, for those of you who might want to work with universities or work with academics in universities, it's really important that they create the right paper trail. Careful planning, because if you don't, you can really mess up and get yourself in a lot of trouble. Get it in writing. Luckily, somebody taught me that very early on. Get it in writing. Transition from NIH funding, know when it is that you should take something forward. And luckily here, I was able to learn a lot of this from my two former professors, right? So one of my professors, as I said before, was Len Herzenberg. His technologies ended up being a multi-billion-dollar industry. Len's lab both created the flow cytometer, which in and of itself was the top grossing patent at Stanford for a while. And then out of his lab as well came a technology called humanized antibodies. You probably all heard about the antibody injections that people give for a range of diseases: cancer, immunotherapy of cancer, immunotherapy of autoimmune diseases. That technology base came from Len Herzenberg's lab. Len was the consummate academic, but he was savvy enough to know how to implement it and get it into the commercial stage without involving himself personally in the commercialization of it, right? Staying outside, being the advisor, not necessarily the implementer, brought in hundreds and hundreds of millions to Stanford and is as I said before, a multi-billion-dollar industry in and of itself. So he understood what he could do within Stanford and then what needed to be taken outside. So for me, at least, when you still want to use that technology or when you're still in that betwixt and between stage, the idea is that Stanford and many universities get very worried that you're a technology pipeline to the company. They just will not allow that, and they're right not to do so. Because that's when students and postdocs start getting paranoid that their ideas are being lifted by the company, right? So how did we manage this again and again? So again, for me, it's relatively straightforward. We were creating a technology base, a technology foundation. We give the technology foundation to the company to perfect, work out the bugs, make it so that it will work from one side of the planet to the other without somebody standing over it minute by minute, right? So we keep the technology QC at the company, right? But we use the technology at the university, right? We use it for what it's designed to do, to discover new things around cancer, around immunity, around whatever it is we're interested in, but we keep the technology QC and the development of the instrument or the reagents at the company. And by doing this, we can satisfy the requirements of the deans that if there's anything new that's developed at Stanford, it would then have to be appropriately licensed again by the company. No technology pipeline, just can't happen. Because just the number of things that can go wrong are innumerable. And so, as I said, be very, very careful of the wall between the ideas at the university versus the company. So, really, it sort of ends almost with this. Now that you have a company, what's the role still of an academic? As I said before, not all the VCs are created equal. They'll see your role depending on their personal biases. Many of them almost begin with asking the question, well, are you going to move to the company? My immediate answer is no. I'd probably be fired in about 10 minutes because I don't like people telling me what to do. So not all VCs like that, right? So how is it then that you can act as an advisor and keep your distance, right? That's something that just has to be learned, right? Because it's your baby. When things are going wrong, you want to insert yourself into the process and become to fix it, right? And often companies, well, not often, always companies build up their own internal personalities and managers, and they don't want the academic coming in and vetoing them every 5 minutes or questioning them, right? Because you're actually taking away their authority. So there's a balance there that needs to be learned. And of course, everybody here who might be a business person understands finding the right management is key. But from the academic standpoint, you have to learn to delegate that authority. And again, work with, not against whoever it is. And as well, often you feel like you want to be on the board. I found that actually it's almost better to be a board observer In fact, my first two companies, I made sure that I was only a board observer because I already saw the politics happening on the board. I didn't want to have to be a voting member. And also from a liability standpoint, not being on the board when you don't really understand what's going on is a good thing because you're not basically suable if something does go wrong. So I try to tell my academic colleagues that be a board observer, let the experts really run the voting unless you feel that you're so important that you need to vote, right? But usually they're going to ask your opinion anyway, right? So again, that comes down to a level of trust. Can you trust the VC? Is it the right kind of environment? And as a founder, knowing when your role is over, right? Knowing when to walk away. And as Lee Herzenberg, the wife of Len Herzenberg, also was a scientist, is a scientist at Stanford, is you have to let something go to let something else come into your head. And so letting it go gives your brain the freedom to go on and do something else. And once you have the bug and you know how to start up a company, it actually becomes pretty simple from that point. Though post the company already being started, you need to learn how to promote the technology carefully without being so much of a promoter, you're practically a car salesman, right? So that's you have to be very careful. And the best way I found to do it is just do good science and talk about the science. Don't talk about the company, talk about the science. People will know the technology that got there. Let others do your PR for you and mitigate the conflict of interest issues, people being jealous of you, by sharing broadly. Right? Just give the technology away as much as you reasonably can without, obviously undercutting the company, and give back to the public their investment, charitable works, right? Give back what you've done, and that will really be very well appreciated by everybody. And then you can point to the NIH and others, and then you become known for somebody who gives and gives back. And when the next company idea comes up, they'll go, well, he did it before, he'll do it again. So let's go forward with that. So that's the talk. I hope that gave a sense of where things are for my laboratory and how I've been applying it. We've been focusing in the use of this almost continuously on cancer research. And luckily and interestingly, because we had spent the last 5 years developing all of the reagents and the technology base, et cetera, for implementing this, when COVID came around, it was actually very easy for us to retool the laboratory to focus on COVID. And we've already actually published a paper now on COVID showing how the virus, where and how the virus gets in. We now have basically clinical materials from 30 patients who died from COVID, 100 different tissues from each of those patients that we're applying all of this technology to basically give an atlas and a geography of how the immune system interacts with the virus-infected cells and what the pathological consequences are to the individual from that. And so again, the first thing we're going to do, as soon as we've got the paper ready and we've submitted it, every ounce of that data will go online for free for others to mine, right? Because that's how you get other people to really pay attention, and you're giving back. So that's it. I hope I was on time.
As usual, Garry, you have done an amazing job in getting information across in the most understandable and enlightening way. And the audience here, I'm sure, appreciates hearing the process, and also it's exciting to hear your strategy for doing science, which is number one, answering questions that had not been answered or not even knowing how to answer them, taking that next step, asking the right question, and finding a way to get there. Secondly, the sharing part is extraordinary. It's something that the Samuel Waxman Cancer Research Foundation is known for collaboration and sharing of information. We've been doing that for over 40 years. And that's the way you can get networking going to make things happen. Motivation of younger people, as well as your colleagues, to go forward with ideas. And you were a good example of such a person when we first met, when you were doing CyTOF imaging 10 years ago, Gary, more or less.
Yeah.
I mean, a novel idea that you were ready to share immediately. And you made it available to all the scientists that we were funding, including my own lab. And so you're right. The way you get an idea across is creative, but you also share it. And that makes it a successful venture. That's a terrific approach. The giving back is a really important part of it. I guess you're not the usual person when it comes to that. Somehow it's come to you probably early in your life. The idea of giving back comes with some degree of success, but it probably started very early with you when you gave back to your students. You wanted them to be successful. That's giving back, not taking away from them, which I admire. But that's now led to, for example, your relationship with the foundation, where you have initiated giving back, and continuing to grow that idea will be very helpful to us and other charitable organizations that work real hard to give people like yourself seed money to take an idea and go to the next level. So that's another entity. Now, I haven't seen any questions that have come up, but I'd like to ask you one question, which we've talked about briefly. The imaging that you show at different levels, the neighborhoods, as you know, an interest of the foundation is to understand why aging is the major risk for cancer. So how can we ask that question by looking at the neighborhoods that, like in colon cancer, and start to ask the question, are they different in inflammatory disease with aging, and in cancer with the same type of markers that you're using? Have you started to look at that?
So that initial cohort that I showed you of the 750 colorectal patients, which we culled down to 34 patients, because we took the extreme examples, right? In science, it's often best to play off extreme examples against each other to prove a point. And they were as best as possible age matched and disease matched. But now that we've proven the point that there were differences, and there are important differences there, now that's where the next step is what you're just pointing out. Now we can go back and ask a different kind of question from the same original study group to say, okay, well, we have colorectal cancer patients who are in their 20s versus those who are in their 60s and 70s, who have, let's say, the same kind of disease. But the open question is, are there differences in their immune systems, right? In the neighborhoods, is there a key neighborhood missing, right? That might be the linchpin neighborhood that makes all of the rest of them work together just the right way, right? Those are the open questions. We already saw an example of that in the study that we did, where there was a particular neighborhood. It's a granulocyte enriched neighborhood. It means nothing to most of the people on the call here. This granulocyte enriched neighborhood, if it is present in the patients who do poorly, as a general group, those patients with that neighborhood do better, right? So that one teeny neighborhood that you just can't see with any of the other technologies that are out there, if that is better, you can go to that patient and tell them, you know what, your prognosis is not as dire, right? You're going to do better than the rest of them, right? So that, I mean, okay, so this is something that I've shared in many talks. I've had cancer myself 10 times, right? Five basal cells, actually 11 times, 5 basal cells, 5 malignant melanomas, and kidney cancer, right? Always luckily caught early. And so I didn't have to have chemotherapy. So I know what happens when the doctor tells you you have cancer and here's your prognosis, right? So but the moment you're given a number as to what your chance is, your mind immediately goes to Las Vegas. What are my chances? Do I win or do I lose? So every increase in your chance of living is a psychological boost to your, frankly, outcome, right? That it's important to know those things. It's not just, hey, I found some little interesting feature about this cancer. It's if you can translate it immediately into an actionable piece of information for the patient or for the doctor to give a new therapy, that's the important part. So yes, we can go back now, get round out to your question. If we can go back now and do a longitudinal time study on that same group of patients to see, well, what is it that's different? And again, we can go back to today just to give it a sense of urgency today. Why do COVID patients who are young get better than, or quicker, or don't get as sick as those who are older? What's the difference in the immune system states, right? Why do some young people get sick and others don't? We're using this technology exactly for those same kinds of questions. There's something about older immune systems that is important that leads to cancers, but then it begs that question. Well, if it is bad to be older, what is it that they're missing and can we give it back, right? And so as it turns out, this technology, we can build models of how the cancer is operating that actually point to both the Achilles heels and mathematically allow us to find what it is that's missing. So that's, I think, the exciting part of how to work with this.
Indeed, it's exciting for all of us. We're getting close to the end of our time. Michael, do you have anything you'd like to comment about at this point?
Yeah,
There's a question from Howard Kurtz as well. Go on.
Go ahead, Howard.
Oh, I can ask it. He says, when applying for NIH grants, for example, do they normally request a royalty or economic interest in any commercialization of research they funded? Do they place restrictions on the IP? No, there's a law actually and a policy where the NIH, although they will own the IP ultimately at some level, they actually grant to the university the right to own it and then implement it, right? So while the NIH, I suppose, could take that back, you don't, I wouldn't want the NIH basically messing in my IP discussions. Better to do it locally, let the universities handle it, which they often do quite well.
Garry, I have a question since you brought up COVID. I'm sitting at my desk here in New York and I'm looking at the headlines as they come on the tape. Moderna with in phase 3 trials injecting 30,000 patients with antibodies, and they're expecting positive results. Any thoughts on how to think about that and how long the antibodies actually could stay in our system?
Yeah, I think the issue here, and this is, when you get infected with a pathogen, you get a boost of your antibodies that are specific to that pathogen. It is natural that in the weeks and months afterwards, the levels of the antibodies will decline. The issue is not how much antibody you have, it's whether or not reintroduction of the pathogen creates a faster response by the immune system, what's so-called memory, right? So what has not yet been proven or disproven, and everybody's running around with their hair on fire as if dropping antibodies is something that's bad. No, it's good. If you kept the level of antibodies in your blood at the peak as from when it was that you were first infected with the pathogen, your blood would be sludge with antibodies of the thousands of pathogens you've seen over the decades, right? It has to go down. But the immune system has memory. It's poised to reactivate much more quickly with both antibodies and what's called a T cell response. And to flip that back around as well to cancer, it's probable that many individuals have created their own antibodies and their own memory responses against cancers that they've had in the past but never even knew it, right? So the open question is, how do we teach the immune system either not to forget or not to allow the cancer to force it to forget, right? Because that's one of the tricks that cancer does, is it leads to loss of memory of what it is that you should do. So maybe that's one of the things that happens in aging, that as we age, we forget we forget how to remember, right? And so that might be one of the key things that we should be designing new studies to ask questions about. Why do we forget to remember? Right? Or how does the cancer step in and interfere with our memory response?
That is a terrific biology new idea that I'm challenging you to take forward.
Yeah, well, you know I am. So,
and we certainly will work closely with you to make that happen.
yeah, well, thank you. Your support was really important. It really was.
Right. So I thank you very much, Garry. This is probably one of the best webinars we've had. The audience is really been appreciative of having this opportunity. Michael, thank you for introducing it and leading us as we go forward. And Garry, we look forward to a lot of future interactions in the future.
Same.
Thank you very much.
Thank you,
Stay
everybody,
well,
for
stay safe.
Thank you.
Okay.
listening.
Be
Okay,
well,
bye-bye.
everyone.
Right. Bye-bye.
Bye-bye.
Mhm.
Thank you for attending. The webinar is now over.